Hi, this is Let's Discuss Soon after reading it. We are selected and read the paper about consciousness, and from now we will discuss it. Firstly, we will introduce ourselves. Please, Yota.
Hi, I'm Yota Kawashima. I'm a research assistant at ATL and Monash University.
Thank you, Yota. And me, I'm Ryoichi Watanabe at Kyoto University and a PhD student. Then I'll introduce today's paper. Today we will discuss the title The Shape Perception via High Channel Count Neuroprosthesis in Monkey's Visual Cortex, written by Xian Shen, Fan Wan, Eduardo Ferranzedi, and Peter Loesema by Science and 2020. Firstly, I will introduce briefly summarize. So in this study, the authors used so many electrodes in the intracortical for two macaque monkey and to stimulate like the primary visual cortex and to produce some phosphone. And as phosphone, they made the like a simultaneous stimuli to create vertical and horizontal dot percept and a sequential stimuli to create stimulus movement like horizontal and vertical movement of dots. And also then the authors made the letters or figures perception like the L or T. And then macaque monkeys are, how to say, report their phosphone, like the particle, if the particle is happen, monkey suckered is under or upper or something. So macaque use the suckered response for the phosphone. And the result showed that the macaque monkeys can or could perceive and report the phosphone made by the intracortical stimulation. No, like the dot appeared or dot movement or dot is very vertical or horizontal, or the stimuli was like the T or L. Yes, and this is a result. And there was no running effect, and means that the first in the test trial means trial 1 to 10 and trial 21 to 30. There's no difference. So these are results suggest that how to say we can make the complex experience or complex phosphone by using the brain stimulation. And so in this study will contribute to the medical context or something. And I think this advantage point of this study is that the authors used the 1024 is a high spatial frequency and intracortical stimulation. And then, so macaque monkeys can perceive and report successfully the figure or shape or spatial information or moving information or letters information. This is my briefly summarized. So, and if you have any comment or addition, please, Yota.
Yeah, I mostly think understand your summary. So this is basically like showing that how we can restore vision of macaque by microstimulation with high or fine scale intracortical implants. And I think another advantage the author mentioned is that they can or they did recording from V4, and then they claim that by monitoring the activity in V4, we can determine the stimulus threshold for phosphine, and by doing that, we can skip the time-consuming calibration procedure that require behavioral reports from subjects with many trials. So that's I think the another advantage they claim in this paper. So the recording from V4 can determine the threshold for the stimulations.
Okay, thank you. Yes, V4's information is important in this study. Okay. So, and do you have any question or confirmation of this study?
No, I don't have any. I mean, yeah, the paper was clear. It was really, easy to follow. Then result was, easy to interpret. So nothing, no question.
Okay, yeah, me too. So, this paper is so clear. So let's discuss our thought or interpretation. What do you think, or do you have any questions or comments?
Yeah, I understand, it's very difficult to restore vision by microstimulation. And then what we can do is just, create the phosphine, which is just, I think, or to be honest, I have not experienced the phosphine before, so I don't know how I feel, how I experience it. But in the paper, they mentioned that some phosphine are colored depending on the stimulus locations. And then, basically by mapping some, or creating a mapping between the, cortical area and receptive field, which is just a retinotopy, we can, create the shape by, creating the pattern or some specific pattern of stimulation. Like in this, study, just a letter L, T, and something like that. And maybe we can, with this technique, we can make more complicated, patterns, such as I don't know, like a square, circle, something like that. But I think it's really interesting, or I don't know how we can do it. But then if we can do induce some color perception, it will be very interesting, but I understand it's very difficult. Well, yeah, that's just my thought, not even, I think, questions or, comments to this paper.
I see, yes, so, yeah. So color phosphon, how do we make the color phosphon?
Yeah, but if I remember correctly, color is encoded in B4.
Yeah.
Right. So my question is that, okay, by stimulating neurons in B4, can we, experience some specific color
Mm,
or just stimulation before is not enough?
I see.
That's my, kind of questions.
Mm. And I think there is a color pathway at the primary visual cortex, visual area.
Mm.
So previous research stimulated not only the previous research or how to say, the limitation of the human studies that the more or less frequency, spatial frequency, right? So electrode is the number of electrode is like more or less than this study or the macaque study. So so spatial frequency was so low. So I think the color pathway was included by the stimulated field or something.
Mm.
Mm. Yeah. So yeah, interesting. And also, like the fusiform GRS or face area or more higher order area, if we stimulated this area, can we made some faces or humans, like the images or something?
Yeah, now I remember that I read something about that, which is I think implant for human, not monkey. So I think for epileptic, patient, then if I remember correctly, the study was, presenting some circle or the ball to the participant, the subject, and they stimulate some brain region and then ask what, subject perceive. And when they stimulate the fusiform area, they perceive something like not face, but not clear face, but something like face, if I remember correctly. Yeah.
I see. And the good point of the visual first area or primary visual area is that freely stimulated. We can stimulate freely. It means that we can use the retinotopy, so we can simulate or make the variety of visual information, right? But in the higher order area, like the more abstract, how to say, information or so face or object or something. So we can use freely simulate the visual experience at the primary visual area by using the retinotopy. Right? So interesting.
Yeah, I'm now searching for the paper I mentioned, but I'm not sure that I can find it during this conversation. So if I find it, I'll share it to you.
Yeah.
And yeah, another. Yeah, but anyway, this paper was really interesting. And I can see the development of technology, like with this, huge amount of, channels.
Yeah.
Yeah.
And so, can I say my question?
Yeah.
And so I'm wondering which is better, like the sensory substitution or sensory transition, means vision from vision to tactile information or something, or brain stimulation in this like this study. So I'm wondering which is better, the transformation of the transformation of perceptual information before the input brain or after brain stimulation. Is it clear what you?
Yeah, I think I understand your point. So this is one, way to, or the participant or the subject feel the vision.
Yes.
But instead, we can convert some visual information to, let's say, auditory information or tactile information. And then let's say talk about the tactile information, but maybe the auditory information will be much clearer because if we have some camera, right?
Yeah.
And then that can recognize object, saying that okay, in front of you there is table or there is chair, something like that. And then if we attach some speaker to the camera, then it will speak out. So there no need for participant to be have micro stimulation directly to the brain. Yeah, I understand that your point. And this is invasive stimulation. This technique is invasive, so of course it will hurt to some extent. It will hurt the brain to some extent. So I'm not sure which is better. But one thing I can say is that again, I have no experience this feeling of phosphine. But if I don't know, but this technique will generate some sense of vision, right? You feel visual experience. But if you transform or replace some visual information into another sensory information, yeah, you can still have necessary information about the environment, but you lose the information or you lose the experience, right? The visual experience.
Yeah.
So that's I think the difference. And some people say, okay, I don't need experience, just information or the knowledge around the world is enough. But the other says that, oh, I want to feel the or vision, something like that. But I don't know. And that's one thing. And another thing, yeah, I don't know. In terms of the cost, I would say that this technique will cost a lot. I mean, money and time, right? But if you have camera and the speaker, and current deep learning, machine learning technology, yeah, it's still not perfect. But we have auto driving car, right? So we have some technologies that will be at least enough only for driving. So yeah, I don't know. So in terms of the cost, yeah, the alternative techniques might be more beneficial over the brain stimulation.
Yeah, I see. And also, yeah, this is a cost that the how to say, yeah, the brain damage is also happen. This this approach. So this is also the cost.
And you need to take surgery.
Yeah.
And I don't feel no one will, prefer taking that. Yeah, so I'd say, yeah, there are many factors. And we also need to think about the personal preference. Yeah, I mentioned that the experience versus just the information or knowledge. Yeah, but at least, having said that, at least, showing that we can do this is very important. So yeah, in that sense, this paper was really, interesting and
yes. And also, I think the how to say, experience or brain development is important, right? So after of accident or something, people cannot see, but before of the accident, patient could see, and the visual area is developed. Is important. So then, this technical approach is good. But how about the how to say, before of the birth, people or infants can so how to say, before of the birth, person cannot see. So who cannot see by the damage of something eye or visual pathway, and then the visual area was not developed. Then this simulation approach couldn't be used.
Yeah, I totally agree with your point. If participant or monkey were blind from birth, then we're not sure whether the brain has enough capacity or the some processing for visual informations. And also another point is that in order to kind of design the pattern of stimulation, and we need retinotopic map. And in order to get that, we need to do experiments, right? But well, I'm not sure whether how variable the retinotopic map is across the individuals. But if it's different, really quite different, then we can't apply this without having or before having retinotopic map for that individual participant. Otherwise, let's say if we want to present some vertical line, so we design based on design that stimulation pattern based on others retinotopic map, but still it's will be some distortion will be there. So maybe it looks like a little bit inclined or something. So yeah, it's gonna be very difficult. We can't get that retinotopic map for that subject. Yeah, I agree. And yeah, your point is, I think, very reasonable. Whether we have or we can still get this phosphine for blind people who have that or who is blind since they were born. You mentioned that development is very important, and experience dependent some learning will be very important, right? Yeah, so I agree with your point. Yeah. Yeah, oh, yeah. Okay. Yeah. I was, going to, I was saying that, maybe how about other modalities, not the visual, but, audition or, tactile sensation. But I think audition, it's more because implant cochlea is, I think, is widely used technique. I think I don't know the underlying mechanism for that, whether it's directly stimulate brain or just stimulate some cells, neurons, or receptors in the eye, not the brain. Not sure the mechanism there, but yeah, because vision, because we have retinotopic map, we can design this pattern. But for, let's say, audition, can we have this kind of something like retinotopic map for vision? Or maybe for tactile sensation, we have more clear map in the brain, somatosensory map. Let's say this area is for hand, this is for face, and things like that.
Yeah, so Penfield is one. Penfield's brain mapping.
Yeah.
I see. Yes. So like the artificial inner ear. Yeah. So this study is so contribute to our life or so. But I don't know how about the human study or recently human study. How to advance from this study, this paper? I don't know, but
yeah. Yeah, intracortical implant. That part, I am not sure whether we can, directly or apply this technique to human subject in near future or not, because of ethical problem. But I don't know. But there should be some cases for this kind of study or clinical practice. No, I think because otherwise no one can record the activity, electrical activity inside the intercortical activity, like I don't know, LFP. But no, yeah, I think this paper mentioned that the size of a brain as well.
Yeah.
The size is small. The brain size is small for monkey than human. So if we want to cover the
so
whole brain area, then we will need more electrodes.
Yes.
That will, it will more likely to cause problems if we use more channels or the electrodes. So
they also
but yeah, I think that's all for me. Again, this paper was interesting. And I feel, or I kind of understand how difficult this will be. Like at first we need to, or they need to develop the electrode with this many channels, then do experiment. So it'll be really, I think, hard work, and again, this work is really great.
Yeah.
If nothing else, Ryuichi, shall we, wrap up here?
Yes.
Okay, then see you next week. Goodbye.
See you.